So let's look at how we use our linked genes to determine how far apart our genes are. So we've already established that if there's no crossing over between two genes, we don't get any recombinants. When crossing over does occur, most of the offspring are going to be the parental type, but some of them are going to be recombinants. Now, not all of them are going to be recombinants because remember, these are separate meiosis events that are producing each one of our offspring, and crossing over doesn't always happen in exactly the same spot. So crossing over may not always happen the same location between the same genes in every gamete that is produced. So only some of them are going to have recombination events between the genes that you're looking at. So here we've got tomato plants. We've got normal leaves and tall plant with a mottled or speckled leaves and dwarf plant. And then our parental types are going to be the normal and tall and the mottled and dwarf. And if we look at these numbers here, we have about 50 or so each, quite a few compared to our recombinants, which are less than 10 each, so less frequent. So we can use the total number of recombinants to give us an estimate or a relative difference of how far apart these genes are. To calculate our relative difference, we are going to calculate the recombination frequency. Now, a general formula for a frequency is always going to be the number in a subset. So whatever our subset is, divided by the total number that we're looking at. So for example, when you take your percentage for a grade, this is a frequency. You're looking at how often you got points out of the total. So this might be 85 points out of 100. And then we're always going to convert our frequencies into a percentage, so we're going to divide it by 100. A frequency should never be more than 1, a full value of 1, and never more than 100%, because we're always looking at a subset. Now that subset could be the whole, it could be all 100 out of 100, which is equal to 1 or 100%. Or it could be a small subset, let's say 75 out of 100, which would be 75%. So in order to calculate the recombination frequency, we need to look at the number of recombinants divided by our total number. So we learned that Bates and Punnett and Saunders showed that sometimes these genes are coupled or don't separate independently like we would expect. And then Thomas Hunt Morgan showed us that genes are located in specific places on chromosomes. And this, of course, both support the chromosomal theory of inheritance. So we learned about this in chapter 3. This basically says that genes are physically located on chromosomes. There are a few more things that are involved in the chromosomal theory of inheritance, but that's the tenet that is important right now. So our recombination frequencies are going to allow us to look at the proportion of offspring that are exhibiting these genes that are found on these recombinant chromosomes. So let's look at an example. We are going to cross two cucumbers, and their genes are for the texture, either warty or smooth. And then their appearance, either dull or shiny. So we have 4 options here. We've got the parental options that are warty and dull and smooth and glossy. And half of our types are going to be parental types. The other half of our types are going to be the recombinant. Now the recombinants then are going to be the warty and glossy and the dull and smooth. So if we look at our progeny here, we can see that there are approximately 42 warty dull fruits, and there are 42 smooth and glossy fruits. So these are the parents, and we mentioned that parents are the most frequently occurring. So this is 42% of our offspring are one parent, 42% are the other parent. If we look at our recombinants down here, 8% are the warty and glossy, the first recombinant, and then 8% are the smooth and dull, which are going to be our recombinants. Now we can use this information to calculate the recombination frequency, or how likely it is that crossing over is going to occur between these two traits. So to calculate our recombination frequency, or the percent of offspring that are recombinants, our recombination frequency is going to be the number of recombinants divided by our total. So we don't have an actual number here. We know that 42% of our individuals are one parent type, the warty and dull. So we're just going to assume out of 100, that means 42 individuals. 42 individuals for other parental type, 40 for each of our recombinant. So to calculate our recombination frequency, we're just going to add up our recombinants, 16 out of our total, which we are assuming is 100, which is equal to 0.16, or 16%. So for this particular set of genes, our recombination frequency is 16%, meaning that 16% of our offspring are recombinants. So what does this have to do with our eukaryotic gene mapping? Well, we can use this recombination frequency to give us a relative position of our genes on a chromosome. So we call this a genetic map, and historically this was the first type of map that we had. And what would happen is that researchers would go along a chromosome and they would count recombination frequencies between two genes. So they would find out relatively how far apart they were. And then they would do it for two more genes, another gene, let's say we've got D in the middle, and they would just move along the chromosome looking at recombination frequencies. So this was really tedious because you had to do a ton of crosses. You had to do enough that the statistics were going to work out. You needed a big sample size, but this was the best that they had. Now we can do a variety of different types of maps. We can do what we call physical mapping, which is where we do DNA sequencing and we look at the base sequences, the specific bases, and we can look for patterns in our base pair sequence that indicate to us that it is a particular gene, or we can look for homologous sequences with known genes and kind of figure out where genes are. And that's going to give us a specific nucleotide address. So that's going to give us like the actual real physical position. On a genetic map, it's all going to be relative. So we might know that A, B, C, and D are close to one another. It's not really going to tell us exactly where it is on a chromosome unless we can do recombination frequencies all along that chromosome. So it's going to give us information compared to other genes, and it'd take a lot of work, totally possible, but a lot of work to get the actual position. But this is how we did it. This is how we mapped chromosomes before we could do sequencing. And we were able to get some really detailed maps this way. So the distance between our individual genes based on our recombination frequency are called map units. So map units is kind of like saying, if we say it's 10 map units apart, it's like saying it's 10 inches apart. It is not like saying it is 10 nucleotides apart. It is a distance, a relative distance. So we call them map units or centimorgans after Thomas Hunt Morgan. You may call them whatever you like. If you want to call them map units, the abbreviation is MU. If you want to call them centimorgans, the abbreviation is C with a capital M. And we'll talk about map distance and map units a little bit more later. We are going to stick with what we call map units for now. A map unit is going to be equivalent to a percentage. So 1% recombination is equal to 1 map unit. This is slightly different than a map distance, and we're going to be using map distances in the lab. Other than that, we're going to stick with our map units, just so we're all on the same page. And I'll explain in lab why we are using map distances and not map units. But a map distance is going to be the recombination frequency divided by 2. And that's just because there are actually 2 possible recombinants for every crossing over. So we're saying, well, we're recognizing that yes, crossing over has occurred, but we're actually getting 2 phenotypes from 1 crossover. And so again, map distances are just a little bit different. We use them in lab. If I don't specify, you're going to just assume you are using map units, and we'll just go with that. All right, so our genetic distances are approximately additive. This means we can use multiple genes and their recombination frequencies to tell us the relative distances. So I'm going to do the first one. I would like you to do the first one with me and then try the other 2. And then once you do the first one, go ahead and pause the video, try the other 2, and then I'm just going to write out the answers. And you can check your answers and then come see me during office hours if you have questions. So for the first one, we are looking for the location of A, B, and C. And we have a little bit of information. We know that A and B is 5 centimorgans apart based on recombination frequencies. So this would be a 5% recombination. B and C is 10 centimorgans apart, and A and C is 15. So you could just start drawing this and see if you could make it work. Or what I like to do is look at the ones that are farthest apart, because those are going to be the ones on the outside relative to the other. So if we know that A and C are 15 centimorgans apart, and we also know that A is closer to B than C, and we know that B is 5 centimorgans from A, we can place A a little bit closer. And then we see that B to C is 10 centimorgans. So our order is going to be A, B, C, and we can see those map distances. Now, if we add the distance from A to B and B to C, it should be roughly equal. They will not be equal every single time, but they're going to be close, because I made up this data, but for our other data samples, they're going to be real life recombination, real examples from studies that people have done, and by chance, we may not get exact, but we're going to see that our numbers are close enough that they're roughly additive. So go ahead and pause the video, try the other 2, and then come back and check. When we use recombination frequencies for a linkage, there's a couple of things we have to keep in mind. One is that if we have a 50% recombination frequency, this is essentially the 1-to-1 to 1-to-1 ratio, telling us that 50% of our offspring are recombinants, which means that these alleles are assorting independently. And this is exactly what we would expect if our test cross was showing independent assortment. So 50% recombination means that our genes are unlinked. What that means is they are not part of a linkage group. So that means that they are not located close together on a chromosome. It means that they're either located on opposite ends of the chromosome, or it means that those genes are located on completely different chromosomes. Recombinant mapping can't actually tell you if it's on the same chromosome or just on the other end or on a different chromosome. You have to do lots and lots of recombinations all along to figure out if it's actually on the same chromosome. You basically have to map both ends. So we know now that 50% recombination frequency means they are unlinked. And that can tell us that they're not nearby. We also want to keep in mind that there can be two crossover events between a particular set of genes. If they are far apart, enough apart, then crossing over can happen twice. And what's going to happen is that the red chromosome is going to switch with the blue one, and then it's going to switch back. And you don't see these as new phenotypes. These are not new phenotypes because a switch has happened, but then the switch has gone back. And so yes, there's a switch in the center, but we don't see it because we're looking at the phenotypes for the A and B genes. And so that can also happen. But we can use that double crossover event to tell us the location of 3 different genes on a chromosome, or the relative distance of 3 different genes on a chromosome. And this is called a 2-point test cross. So we're going to use our recombination frequencies for a couple of different things. One is to tell us if they are linked or unlinked, and then the other is to give us the relative distance of 3 different genes. So let's do an example. We have a set of 4 different genes here, A, B, C, and D. And we want to figure out which ones form a linkage group and then a little map of our chromosome regions. So we need to find out which genes are located closely together in a particular group. So we're going to look, we've got A, B, C, and D, and we need to put these in groups. So if we look at A and B, there's a 50% recombination frequency. That means they are unlinked. So A and B are going to be in different linkage groups. If we look at A and C are also unlinked. So that probably means that C is going to be linked with B, but not necessarily. We need some more information first. A and D are also unlinked. So A is not linked to any of these other genes. If we look at B and C, their recombination frequency is less than 50%. So B and C are going to be somewhere linked together. B and D are also less than 50%. So we know that B and D are close together, and C and D less than 50%. So we know that they're close together. So now let's use this information to tell us how far apart B, C, and D are. So I like to use the 2 that are farthest apart. So C and D are the farthest at 28, we'll call it centimorgans. And we see that B is closer to D than to C. So let's go ahead and put B here. The distance between B and D is going to be 10. The distance between B and C is a little bit longer. It's going to be 20. Remember, these are roughly additive. So while not exact, 20 plus 10 is pretty close to 28. And so we know that we've got our right distances. So here's another problem that asks you to look for linkage groups and then to map each linkage group. So keep in mind that recombination frequency of 50% means that genes are unlinked. So that means they are on different linkage groups. I'm going to do the first 2, and then I would like you to pause the video, See if you can figure out how many linkage groups there are, and then make a map. And then I'm going to go ahead and work through the problem after a brief pause so that you can see what happens. So if we look at A and B, their recombination frequency is 10, which means they're pretty close together. So they're going to be on the same linkage group. So I'm just going to write A and B up here. If we look at A and C are on different linkage groups because that recombination frequency is 50%. So we're going to put C down here for now. If it is unlinked with A, it is probably going to be unlinked with C. And if we look down here, B to C is also 50%. So that corroborates the second linkage group. So go ahead and pause the video, and then I'm going to finish working it, and you can come back and watch it when you're finished. All right, so if we keep going, A to D is 14, so that means they're pretty close together. A to E, 50%. So that means that they are not on the same linkage group. So I'm going to put it down here with C for now. And then A to F, 50%, so unlinked. So I'm going to put it down here. A to G, 50%, so G is down there. Now we're going to move on. We're just going to keep going. B to D is 4. So we know that B and D are in the same linkage group. We already knew that because A and B were linked. B to E is 50, so that means it's in a different group. B to F, also 50. B to G, also 50. So that just checks with our A information. C to D is 50, indicating different linkage groups. C to E is 8, suggesting that they are in fact linked down here. C to F is 50. So that's a bit of a problem. We've got it down here on this linkage group, but in fact it is unlinked with C. So we're just going to move it to a third linkage group for now. C to G is 12, linked together. D to E, 50. D to F, 50. E to F, 50. So that again tells us that it is in fact unlinked from C and E. And if we keep going, we're going to get down. E to G tells us they are linked, and G to F, they are unlinked. So we have 3 linkage groups. We've got the A, B, D linkage group and the C, E, G linkage group. And then we've got the F linkage group. Now this just means that F is unlinked with everything. We can't really do any mapping for that. We just know it's somewhere else. So where we put that in the third linkage group really doesn't matter. But now we can look back at A, B, and D. So I'm going to erase all of my marks here. A, B, and D. So A to B is 10. A to D is 14. And B to D is 4. So that's going to give us some information for putting our genes on the map. So our farthest distance apart is A to D. So I'm going to go ahead and put A to D, farthest distance apart. They are 14 apart. B to D is 4. So we know that B and D are closer together. They are 4 map units. And then A to B is 10 map units. And again, we saw that A to D is 14. Excuse me, so that makes perfect sense. Next, we have C, E, and G. So we've got C to E, C to G, and E to G. Our farthest apart are E and G. So we're going to put E and G far apart from one another. They are 18 apart. C to E is 8. C to G is 12. So C is a little closer to E than it is to G. C and E are 8 map units apart. C and G are 12. Altogether, we've got 18. 12 plus 8 is actually 20, but it's pretty close to 18. So we're going to go ahead and go with it. So now we have our map of each of our linkage groups. A, B, D is our relative order, and E, C, G, relative order, with F on that third linkage group. So this problem is a little bit different. It gives us information about the parents and then asks us to determine the arrangement of the alleles and their relative map distances. So our parents are, or I'm sorry, our alleles are a gene on a gene called for color. So we've got black color denoted by big B, or brown color denoted by little b. And then we've got full color, meaning all black or all brown, which is dominant to chinchilla. So chinchilla is kind of like a mottled, speckled, almost like a grayish, kind of makes like a grayish brown looking color because you've got some kind of speckling in with the black or the brown. So we have rabbits that are heterozygous for both traits are crossed to rabbits that express the brown chinchilla. Okay, so heterozygous for full color and black. So we've got black and full color, and then we've got brown and chinchilla. So this is our linkage test cross. We've got the heterozygous with the homozygous. And so our parents are black and full color, or brown and chinchilla. So if we look at this set of data, we can identify which of the offspring are our parents. So we've got black and full color. That's one parent, and brown and chinchilla. Now notice also that the parents are the most common of the individuals. Total, we have 100 rabbits, which makes our math nice and easy. And we want to figure out how far apart they are. So we're going to figure out our recombination frequency, which is our total number of recombinants. So recombinants are the brown and full and the black and chinchilla. So we've got 16 of one recombinant, 19 of the other out of 100 individuals, which gives us 0.35 or 35%. Now remember that 1% recombination frequency is one map unit. So this means that on our chromosome, we've got the color gene, and then which would be the black or brown, and then we've basically got like the solid or speckly color. So then we've got solid, and so they are going to be 35 map units apart. Now it does say determine the arrangements of the alleles on the heterozygous parents. Since this is relative, it doesn't actually matter if you put color or solid on one side or the other. It makes no difference at all.